Biology Role Model Project. Hi, my name is Ray, and today the scientist I chose was Dr. Belinda S.W. Chang. I'll be referring to her as Dr. Chang. Who is Dr. Chang? Dr. Chang is a professor at the University of Toronto in both the departments of ecology, evolutionary biology, and cell and system biology. She's known for her research on the molecular evolutions of opsins, which are light-sensitive proteins in the eye. Her lab explores how small genetic changes can dramatically alter how organisms perceive light. Education and career path. Dr. Chang began her academic journey at the University of Toronto, where she earned her Bachelor of Science in Biochemistry. She then completed her PhD at Yale, and then went to work as a postdoctoral researcher at Rockefeller University. Later, she came back to University of Toronto, where she worked as a faculty member in 2003 and continues to lead groundbreaking research every single day. Dr. Chang's early interests and hobbies. From an early age, Dr. Chang was fascinated by how genes influence animal behavior and perception. She loved exploring biodiversity and how different organisms adapt to environments. Outside of the lab, she enjoyed anything that was related to the environment, whether that was hiking, photography, or traveling. She especially loved going to places which were rich in wildlife and natural diversity. Why do I admire Dr. Chang? I want to go into ophthalmology in the future, and Dr. Chang's research is focused on exactly that type of section. Dr. Chang bridges the gap between molecular biology and the science of vision. Her research has inspired me to pursue ophthalmology, as it shows how understanding molecular mechanisms can lead to medical insight. I also admire her advocacy for diversity in science, and as she's one of the few women of color leading a major evolutionary biology lab. Research focus. Dr. Chang's field is molecular and evolutionary biology, and her main focus is opsins, the proteins in our eyes that detect light. Her big question includes how do mutations in opsin gene change which color or wavelength of light can organisms see? Or another question is, how do animals adapt to different environments like deep oceans or caves? For the first article I chose to go over was the article Recreating a Functional Ancestral Archosaurus Visual Pigment. Dr. Chang and her team asked, what did the visual system of ancient archosaurs, the ancestors of birds and crocodiles, look like? They used phylogenetic models to predict the DNA sequence of the ancestral opsin gene, and then synthesized it in the lab. They expressed this gene in cells and measured how the resulting protein absorbed light using spectroscopy. The results showed that the ancestral opsins absorbed light best at around 500 nanometers, which suggests that early Archosaurus was adapted to dim light conditions. Now let's go over one of the data figures from this article. On the x-axis, we see that wavelength, and on the y-axis, we see relative absorbance. You can see how ancestral opsins peak around 500 nanometers, which differs slightly from modern species. This shift reveals how even small genetic mutations can fine-tune color visions over evolutionary time. The figure visually demonstrates molecular adaptation in action. Now, for the second article, I chose to go over evolution in action, inactivation, and the loss of short wavelength sensitive opsin genes during the diversification of neotropical cichlids. In her 2021 study, Dr. Chang and her student Frances Hausert explored how cichlid fish adapted their vision to different light environments. Using comparative genomics and gene expression analysis, they found that the fish living in murky waters lost some UV-sensitive opsins because ultraviolet light did not penetrate those environments. Clear water fish retained these opsins, allowing them to see UV light. The phylogenetic tree in the study maps which species kept or lost these opsins, color-coded by environment. What she discovered in future directions discovery. Dr. Chang's work has revealed that small amino acids can shift color vision sensitivity and that opsin genes duplications contribute to color diversity in many species. She's shown how environmental light drives the duplication, modification, or even loss of these genes. Looking forward, her lab is developing artificial intelligence models to predict how mutations will affect opsins function. They're also investigating how similar mutations may lead to human color blindness or retinal diseases, helping connect evolutionary biology to medicine. Lastly, reflection and connection. Dr. Chang's research shows that evolution isn't just about fossils. It's about molecules still shaping life today. Her work links evolutionary biology to vision science and ophthalmology, the field that I hope to enter. Learning about her journey has reminded the scientific discovery comes from curiosity and persistence. Her success as a woman of color in a challenging field also highlights the importance of diversity in science. Thank you, and thank you for listening.